Let's humble ourselves for a prayer. Our Father, who art in heaven, hallowed be thy name. Thy kingdom come. Thy will be done on earth as it is in heaven. Give us this day our daily bread, and forgive us our trespasses, as we forgive those who trespass against us. Lead us not into temptation, but deliver us from evil. Amen. Almighty and everlasting God, we kindly want to thank you for this day. Thank you for the gift of life. Thank you for everything you've been able to create. Thank you for our teachers who have stood up to actually come out to help us through this lesson. And we kindly request you that whatever they may put on the platform, we may understand and we may use it to pass our examinations and be greater people in the future. In Jesus' name I have prayed. Amen.
Amen.
Okay, it's over to you, Teacher Arthur and Teacher Eva. One of our discussants is Brian.
Okay, thank you so much, Mr. Kaziba. Good evening to all the students. I hope you are doing well. Can you confirm that you can hear me?
Yes, we can.
Yes, we can.
Okay, that is great then. Well, my name is Teacher Gonza Arthur. I teach biology, and I'm going to be the lead facilitator in today's seminar. But I'm not alone. I also have my co-teacher, that is Teacher Eva Kabayonga. Teacher Eva, could you say hi to us, please?
Good afternoon, everyone.
Good afternoon, teacher.
Good afternoon.
You're most welcome.
Okay, thank you. Okay, so to save our data, I think I'm going to disable the video. Okay. Let me share the questions. Okay, so today we're just going to have a simple discussion, but this discussion is going to be guided by these questions here. Some of the topics, of course, because I'm sure, I know we have some senior 5 students here, and we also have the majority must be senior 6. But however, the questions that we're going to discuss cut across. I'm sure even the topics, these topics have already been covered in senior 5, most of them. But however, the aim of today's seminar is to guide us understand the best approaches to answering A-level questions. Whether you have learned the topic or not yet, but I'm sure when we are discussing or when we are presenting, we shall gain some skills that we can use to answer all other related questions. Like I always tell my students that when we are discussing the questions, or even when we are discussing maybe exams after writing them, it is not that the aim should be to cram the answers. No, the aim is to see the answer, understand how that answer was constructed, look at the question and challenge yourself whether you could be able to answer that question, or maybe if you attempted to answer it, where could you have gone wrong, or where could you have perfected? So these are the questions that are going to guide us today. I selected them purposively. Question 1 is just an essay type question. Then the other questions are graph questions. So the other ones are graphical questions. Of course, time is never enough. I cannot promise that we shall finish all the questions in this document here. It is impossible, even if we wanted. But however, the few questions that we shall discuss will enable us be able to attempt and also answer the other questions. So where we stop from today, take it upon yourself to also write answers for the remaining questions using the skills that we shall have acquired from the questions that we're going to discuss together. since you got these questions prior, and I had told you to make some research, some written answers, and I hope some of you must be ready. So we're going to start with this question one. With question one, we shall just discuss a few things from it. I want us to use less than 30 minutes on this question here. Then after this, I'll guide you on the do's and the don'ts. Then we go to question number 2. Who could be ready to give us the interpretation of question number 1? If you look at this question, they're saying describe features of carrier-mediated transport across the plasma membrane. And we are seeing the question has 8 marks. Who can give us a brief interpretation? When you look at this question, what does it want? You may be knowing the answer or you may not be knowing the answer, but at least when you read through, at least you know what this question wants. Okay. Yes, Brian? Yes, Louis. Louis, you want to try? Doyle's has, I don't know whether this is a male or a female. The name is not familiar. Doyle's is saying that I think they want mechanisms of facilitated diffusion. So Doyle thinks that they want mechanisms of facilitated diffusion. Okay, we shall look into that. Louis, Bakunda. Okay, Emma.
Excuse me, teacher. For me, I think.
Yes, Louis, you can go on.
I think since they're talking about carrier-mediated transport, I think we have to talk about the carrier proteins, which are actually in a topic we call cell physiology. What we know about carrier proteins is that they have actually, first of all, they have no channels but molecules or ions to get hold of it, and it changes shape in a way that pushes a substance across the membrane. Well, now I think they want us to talk about the different features within that carrier-mediated transport, of which we'll have to talk about partly about carrier proteins and like features and how it does all that stuff.
Okay, thank you. I had the features of carrier proteins. Louis is telling us that we talk about the features of these carrier proteins. Yes, Charles, James. Charles, can you hear me?
Yeah, I can hear you.
Your hand is up. You want to say something?
Yeah, we have just come in. Like we have formed 5 which has told us that we come here. So we have just gotten into. Has it just started or it started sometime back?
We've just started.
Okay. Okay, thank you, sir.
Emerson?
Master, for me, according to me, hello?
Yes, Emerson, go on.
Yeah, if I look at the max allocation, if I depend on the features, I don't know if I can complete the 8. Now, for me, I was suggesting maybe the question requires us to explain the ping pong mechanism.
I beg your pardon?
I think the question requires us to describe the ping pong mechanism.
Wow, the ping pong mechanism. Okay, we shall look into that. Yes, Wandera?
Yes.
Wandera, I don't know whether it is my network which is not clear, but I can't hear you.
Clear, sir?
Wandera, I beg your pardon? Wandera, you're not clear. Wandera, you're not clear. Let me have another person. Rachel. Nakato, Rachel.
Yes, teacher. Me, I think. Are you hearing me?
Yes, Rachel, we can. Wandera, I'll give you another chance later. Let us have Nakato, Rachel now.
Okay, according to me, I would approach that question like I would give the properties of the carrier mediated transport. I would give the properties.
The properties of carrier mediated transport. Can you give us just at least one example of such a property?
Like we have specificity, like carrier proteins, they bind a specific, like they have a specific configuration to bind about, like to, okay, in others they have specific binding sites. So in others they are specific, like there is a specificity like that. I would give in that way. Then another one I would talk about the competition. Yeah. That's how I
What about the competition?
The competition, like when related molecules, like when related molecules maybe they're transported across the membrane by the same carrier. So there, I think there can be competition for another, for the active site. Let me say that.
Oh.
Yeah.
Okay, that is okay. Thank you. Yes, Emma. Okay. Okay. Now, thank you, all of you, for giving in your interpretations to this question. Now, with A-level biology, at first, what is important is you getting the proper interpretation of the question. That is what is most important, getting the proper interpretation of the question. And how you get that proper interpretation of the question? You look at the question and you first try to simplify it. You debunk or you simplify the hard terms that you're seeing there, and you replace them with the easy words, words that you can easily understand. That is always the first thing. You read the question, then if you see there are words that are kind of confusing you, and you know there are others that you can use, so you substitute those ones into that question. Then after simplifying that question, then you start thinking, you start brainstorming of what this question wants with some few examples. Then after doing that, then you open yourself to thinking widely. I've liked Nakato Rachel's interpretation. For her, she's actually, even in her presentation, she's using a term that she's well familiar with, active sites. And she's also using the term that she's familiar with, properties. So for her, instead of saying features, she sees that features are now confusing her. She'd rather say properties. Then where now things seem unclear, she says, okay, think kind of active site. But when you listen to her submission, she has the approach. Now maybe the remaining one is now getting the right ones to use. So that is now the third step, getting the right ones to use. After you interpret the question, you simplify or you replace the tough words in that question, then you now choose the right ones to use. Because remember, this is biology. This is science. Science is science. There are specific ones that we use. If you try to use something different, you may find you're now explaining a concept which is completely different. Like for instance, Nakato was saying that active sites. Yes, the idea behind her using that word active site is actually correct. That obviously there must be a site, and this site must be having a shape, and this shape must be complementary to whichever thing comes to bind in. So that is the idea behind her using the word active sites. But however, that word active site is misplaced when you talk about these carrier proteins. Because you know active sites is just a reserve of the enzymes that once a substrate fits into the active site, it is broken into products. Then these products are released. But however, for such proteins in the plasma membrane, yes, they have these sites, but now we don't call them active sites. So there would maybe disqualify her for using a wrong choice of word, which means something different in science or in biology. So then Lewis was saying that Lewis is saying that we talk about the carrier proteins. Now, when I look at my question, there is nothing like the word proteins. But however, now to him, he's also trying to first simplify this question into his own language. Now, instead of saying carrier mediated transport, he's saying that, of course, he's aware that these are the proteins in the plasma membrane that do this kind of transport. So instead of saying this bulk word, carrier mediated transport, why don't I interpret it that here what they want us to talk about the carrier proteins, which is the correct approach that I'm talking about, that the carrier proteins, how they change the shape. So when you're saying that how they change the shape, then we're now bringing something like a property. You know, a feature is like a property, something like that. So now, what are the actual answers here that we're interested in? Now, we are interested in, first of all, the features or the characteristics or the properties of the transport proteins that are found in the plasma membrane. This is what this question wants. The features, the properties, the characteristics of transport proteins that are found in the plasma membrane. So that is what the question wants. So if I know these features, like actually, Rachel has given us some of the examples, like specificity. So specificity can be a feature. If they say give features of enzymes, they have active sites, like that. Now, Nakata had given us one of the features that they are specific, which is correct. Then she has given us another feature that there is the aspect of competition. There is an aspect of competition in these transport proteins when they are doing their work, like that. Now, after knowing these features, then there is also another word, describe. Because if you say that carrier-mediated transport across the membrane is specific. Now, have you described? Is that describing or that is outlining? That is where sometimes we go wrong and we complain when maybe we find we have scored less marks. If they say describe, if they say outline, then you know what to do. If they say outline, you just write few words like they are specific. That is outlining. But what of describing? Now, describing means you don't give a reason why. But however, you tell us briefly what happens. Just a brief statement of how something actually happens. Then you are describing. If we say that carrier-mediated transport across the plasma membrane is specific, can you describe to us briefly how not the why, but the how part of it. The why part will come in the explanation. Now the description is always asking for the how part of it. Now still using this point here of specific. If I say, if you have somewhere you're noting, you first write down this answer, then we shall see how to use this answer to help us further understand what this question wants. You can say that carrier mediated transport across the plasma membrane is specific, or we can even say is selective. In this instance, the word specific can as well mean being selective. So we can say that carrier mediated transport across the protein is specific or selective. Then now we put there kind of a full stop. Just a moment. Okay, I was still trying to go through what you have in inbox. Okay, so we have that. Now, when you read that answer that you have written down, I want you to read that answer and you read the question again. First of all, is that thing making sense to you? Does that answer make sense to you? When you read through it, is it kind of making sense to us?
Yes.
Can I hear from 2 or 3 people?
Yes.
Yes. Okay. Okay, thank you. So now, it means, first of all, if it is making sense to us, then it means chances are high that that answer is correct. And that is also another thing that you can use to judge whether the answers are correct or not correct. So when you read through it, you see it is actually making sense. Then now, the next step is now describing. Because what we have done there, we have just outlined. Now we're going to see how do we describe. Remember I told you that describing, you briefly talk about how something is happening, how part of it. Yes, Zawadi, you have a question?
Mr. Arthur, I would just like to know, I'm just asking, I thought this kind of question we were supposed to, for example, do first things first. I mean, I thought we were supposed to first talk about the kind of salts, for example, that are too polar or highly charged. We were supposed to talk about where exactly it occurs before we bring in the main features of the facilitated diffusion itself. I would request for some clarity on that.
Oh, well, now, first of all, this question is not asking for facilitated diffusion. That is now a misinterpretation. This question is not asking for facilitated diffusion. Let us start from here. Carrier mediated transport across the plasma membrane. Here, what are we, that in the plasma membrane, we have this kind of transport. There is a way materials cross the plasma membrane. Some materials would just cross by simple diffusion through the pores. Other materials may move across channel proteins. Others may move across carrier proteins. The carriers, they attach on a protein, then this protein carries the substance across the membrane. Now, this question is asking you for only that kind of movement where substances attach on the membrane and get carried across. That is the only thing that this is what this question is asking about. K is saying, but some books show that carrier-mediated transport is the same as facilitated diffusion. Then Amoding Masa is saying, doesn't the word carrier in the question tied to facilitated diffusion? Well, now I'll give you something simple. Now, if I may ask you, does carrier-mediated transport, can carrier-mediated transport use energy? Someone tell me.
No, it doesn't.
Can carrier-mediated use energy?
No, it doesn't.
Now, which kind of transport across the membrane? Okay, where does active transport take place in the membrane?
Across the membrane.
Just across the membrane? Remember talking about active transport, not simple. Yes, Louis?
In carriers too.
In carriers too. Let me just go with that one. In carriers. So it means that carriers can either use energy or they cannot use energy. That is a fact. Carriers across the plasma membrane can either use energy or may not use energy. Now, where they use energy, that is active transport. Where they don't use energy, that is facilitated diffusion. Diffusion does not need energy at all. Anytime you hear of diffusion, that something is diffusing, diffusion, whether facilitated, whether simple, does not use energy at all. So if you choose to take this one as facilitated diffusion, then you are limiting your responses. Yet we know that active transport can also occur through carrier-mediated channels or carrier proteins. Is that clear?
Yes, teacher.
Okay. So anytime you see the word diffusion, that one does not require energy. But however, carriers can also use energy. So when they use energy, then that ceases being diffusion. It now becomes active transport. Okay. Now, someone is saying that what of the channel proteins? Channel proteins, this question now is not asking you anything about channel because we have channel proteins and carrier proteins. Channel proteins, the word channel means they leave an opening in between. A channel is like a route, is like a path, channel proteins. So these ones have a path in between that small materials, they fuse and pass through. That's what they mean. Carrier, something attaches, then after touching, it deposits it towards the inside or towards the outside. Yes, Zawadi? Zawadi, I'm seeing your hand up.
Okay, Mr. Arthur, I would request you to help me throw more light on what exact features are they asking for in this question. Maybe that's where the whole problem is coming from.
Of course, I didn't want to first go to the answer because we have to first learn how to interpret questions. The answers are coming, and also the answers will again help us further understand what this question wants.
Okay. Sir.
So, of course, we have so far, we are constructing our first answer. So far, we have written that carrier-mediated transport across membrane is very specific or very selective. That is an outline of one of the features of this carrier-mediated transport. Now, what am I going to add there to make my point a description using this word describe? So I'll put a full stop and say that specific molecules are carried by specific channels that span the plasma membrane. Write that one as a continuation of the first point that we wrote. You say that specific molecules are carried by specific channels that span the plasma membrane. Now there I have at least mentioned how something happens. How? The description part of it. So that is one answer.
I beg your pardon.
Now someone will have to read first because even I forgot the first words that I mentioned, the exact words. I want someone to read for us the exact words that we have written so far.
You said specific molecules carried by specific channels that span the plasma membrane.
No, you start from where we started with the feature, mentioning the feature. I'm sure we started by mentioning something about the feature.
You said carrier-mediated transport across the plasma membrane is specific, or you can say it's selective. We put a full stop there and we continue to say specific molecules are carried by specific channels that span the plasma membrane.
That is wonderful. So that is our first answer. So you can, when you read through that answer, I'm sure you're now starting to understand what the question wants. Yes, Kay? Kay?
Yeah. Isn't specific or like being a specific receptor a different feature from spanning the cell membrane?
Spanning. Oh. There is some misconception still here. Now, first of all, remember here, what you're talking about, we are actually not giving features of carrier-mediated proteins. We're not giving features of carrier proteins. That is another misconception. What we are giving features of the type of transport that uses carrier-mediated proteins. I don't know whether I'm clear. We are giving features of the type of movement across the plasma membrane that uses carrier proteins. That is what we are giving. Features of the type of transport that uses carrier proteins to move materials across the membrane. That's why we're saying that this kind of transport is specific or is selective. That is the answer that we wrote, that this kind of transport here is specific or is selective, where specific molecules are carried by specific channels that span the membrane. The moment you fail to get the proper interpretation here of the question, then you are completely off. Okay, let me read in the chat. Teacher, we have to state anything like channels in our answers, yet it's carrier-mediated transport. Now, here the channel, the word channel may come in terms of, when we are starting this lesson, Rachel gave us, use the word active sites. So the channel is like that a specific area where this thing has to pass. Now we're using the word channel just as a descriptive word. Then Daphne is saying, teacher, your volume is low. I don't know whether it is low or it is Daphne's network, which is not clear. But I thought it's carriers, not channels. Okay, I think I have responded to that one. Carrier-mediated transport. is the same. Okay, I think I also responded to that one. Then spanning mean that the carriers rotate themselves. Trishila is saying, does spanning mean that the carriers rotate themselves? No, spanning means across. Span means across. Spanning means across. So do they have binding sites as a feature? Now, Japheth Raj is saying that, sir, do they have binding sites as a feature? Now, if you have this in mind, it means still you have interpreted the question wrongly. Remember, the question is not asking you for the features of the proteins. The question is asking you for the features of that type of transport that uses the proteins. Japheth, I hope you have got it. We are not asking for features of the proteins. We are asking her. Who is this one? Okay. Prissy?
Teacher, it's a question. Can we give the fact that it has carrier proteins as a feature, like as a separate feature?
It has carrier proteins, but of course, now that one is still would still lie in the interpretation. Of course, the interpretation, we know it is a fact that they use carrier proteins. So what now the features of that kind of transport that uses the carrier proteins? Okay, now let us look at this second answer. I'm building from Nakato's submission. Nakato gave us that point of specific. Then she also gave us a point of competition. Let me see how best we can use that thing of competition.
Excuse me, teacher.
So, yes?
Teacher, about that point of being specific, what if I say, like, when I'm describing, and I say that carrier proteins bind only molecules of specific shape and configuration?
That is very correct.
Okay.
Okay. Now, let us write this one. You write this one down, then we also use it and we see. There is competition. There is competition. There is competition. I think, let me see if I can type it here. Let me say that there is competition. What's happening? There is competition where structurally Are we all seeing what I have just typed on the screen?
Yes, teacher.
Yes, sir.
Now, read that answer. Do you think it is answering our question? And is now the question starting to make a lot of meaning to you?
Yes.
So that is the logic behind biology. Always read the question, simplify it, write an answer. After writing the answer, then go back and read the question and see whether actually your answer is answering the question. If you write an answer that you also feel ashamed you can't read again, then chances are high that that answer is not correct. Okay, I'll respond to some questions. Yes, Morgan? Okay. VT.
Sir, may have not you.
VT, what is the issue?
Understood the statement.
You've not understood the answer or the question?
The answer.
You read it and read the question again. Read that answer and read the question. Someone using Nokia? His audio is disconnected. Yes, Kalan?
Hello.
Yes, Talan,
Teacher,
can hear you.
Are transport sites the same?
Transport sites the same as what?
Binding sites.
Yes, they are the same. So you can either say compete for transport sites or compete for binding sites. Okay. Then we can now write the third answer and we still see whether it is again making sense. Now the third answer, I will say, you also type it here. I will say, are we reading that statement? There is saturation where transport rate increases as the concentration of solutes increases until all carriers have binded with solutes which they transport. Now here, instead of using solutes, we can use the materials being transported. The solutes, by solutes, I'm meaning the materials being transported. They can be solutes or they can be any other materials that move across the plasma membrane. Now, when you read this answer, the feature is saturation. In the previous answer, the feature was competition. In the first answer that we wrote, the feature was specificity. Then after mentioning the feature, then all these other statements are bringing in the word describe. Because we are describing these features. We are not just stating these features. Now, let me talk briefly how then are we going to distribute these marks. Now, in biology questions, never be scared of the marks. Never be scared of the marks. If the question is saying 8 marks, it doesn't mean that they want 8 points. No. Just rate the biology, interpret the question, rate everything that the question wants. 1 point can even earn you more than 3 marks. Just a single point can earn you more than 3 marks. Okay, so I think I will respond to some of your questions. I want us to use this first hour for this question. Then after that, we go to something different. Okay. Israel Akiki, Martha. What is the question?
Good evening, teacher. I'm requesting you to draw more light for the second point because I'm not getting it.
Now, we are talking about the features of a form of transport. We are talking about the features or the characteristics of a type of transport across the membranes that uses carrier proteins. We are talking about the type of transport across membranes that uses that uses carrier proteins. So we are talking about that kind of transport. And we are saying that in this kind of transport, there can be saturation. There can be saturation. It can reach a point where, when all the carrier proteins have materials attached onto them. So that point we are calling it saturation. Martha, is that clear?
Yeah, thank you, Lord teacher.
Okay. First of all, the moment you think that we are talking about the proteins, then you miss the point. We are talking about carrier-mediated transport. So the key word here is we are talking about the transport, not the protein, but we're talking about the transport. Tony? Tony, are you there? Okay, Tony is not there. Yes, Viti. Okay, Louis?
Teacher, I was asking, so is it okay if you can say that there is binding of large polar molecules such as amino acids, sugars, etc., and undergoing of rapid changes in shape? Is that point okay or isn't?
Yes, so it is correct. It sounds very correct. So you can reconstruct it. How you repeat it? You're saying. Or maybe we can now, okay, let me build on from Louis's point. So Louis is saying that there is binding of large molecules. Maybe I can say that there is bulk movement, or there is binding of bulk molecules. There is binding, let us write down. Let me type it here and we use it as also another. There is binding of bulk molecules onto the carrier, proteins. So Rabakona was saying spinning or rotating, or we can as well say flip-flopping. Oh, I've now lost it. So there is binding of bulk molecules onto the carrier protein, and I don't want to type again. And flip-flopping or rotating to deposit or to move materials across. So you can say to deposit or move materials across. so that it is in this type of transport that uses carrier proteins where we have bulk movement of molecules. So bigger molecules can bind onto these proteins, then they span and get them through. Okay. Now, if I may ask you a question, can we use the point of concentration gradient here? Can we, and why? Can we use the point of concentration gradient here and why? Fred Bamwine.
Sir, Bakunda says polar molecules, are they specifically supposed to be polar?
Not necessarily.
Louis,
not necessarily. They can be any molecules. My question is standing. Are we free to use, also to use the point of concentration gradient? Yes, Vanessa.
I was on my view, like if we use like concentration gradient as in we are limiting ourselves, because concentration gradient is mainly for facilitated diffusion, and yet the question was not like open, so it involves both facilitated and active transport. So if we bring in the point for concentration gradient, we are going to be mainly focusing on facilitated diffusion. So I don't think that will work.
Okay, thank you. Alan, what do you think?
Then you raise your hand.
Thank you, Mr. Asa. For me, I think that point would
Alan, we can't hear you. Alan, we can't hear you. Okay. Well, now, Alan, sorry, we can't hear you. Now, I'm happy that people have now gotten the proper interpretation of the question. Now, when we use that thing of, when you use the point of concentration gradient, we shall be leaning towards the channel. In these ones, carriers, concentration gradient does not play a big role here. Okay, so I think we can go to the next things briefly, about 10 minutes. Then we go to graph work. So now here, the point is not cramming the answers. The point is knowing how do we construct answers. Now, let me go to part B. Now, maybe briefly, if I'm talking about part B, describe the mechanism of phagocytosis in a neutrophil. Yes?
Before we proceed, I understand presentation in biology is also important. So for this kind of question, I'm asking, how exactly am I supposed to present my answers? Do I write in a continuous essay, or I present my answers in short but separate paragraphs?
Okay. Now, Zawadi, if I may ask you, how many points have we written on part
Around
A?
Four.
4 points.
Yes.
Now, have you written them in a bulleted form or in paragraphs?
Paragraphs.
Okay, fine. Now, if we have written 4 points, now these 4 points, you may write them in paragraphs, or you may just bullet them. Actually, bullets in most cases are always better than paragraphs with sciences. You just bullet them. You don't mind even if the point is covering 4 lines, but it is a bullet, it is fine. Now, what of the others? You're free to use full stops. Actually, where you find that you're running short of the correct connecting words to use, I don't know whether they call the verbs or adverbs or adjectives, I don't know. you just use full stops. Full stops. Like for instance, in the first point, which was about the first point was about specificity. Now you can say that they are specific, full stop, continuation. Specific molecules are carried by specific channels, full stop, just like that. If it is 1 point, one point can have 2 or 3 full stops. Where you feel you're stuck of the connecting words to use, like and, therefore, then, where, just put a full stop and move on. That's when you're marking, we look for the marking points. Has this student mentioned about the feature? Yes. Where we see it, we put a tick. Has this student described? Yes, we can see here, she has described. I put there a tick, like that. Okay, so here, describe the mechanism of phagocytosis in a neutrophil. Now this one, I'm sure now here that some people may be off because of the terms used. A neutrophil is a white blood cell. And the phagocytosis, we know that amoeba feeds by phagocytosis. So this question is like asking you to describe like feeding in amoeba. So you describe, but of course you don't say amoeba. Now amoeba here, I'm using, remember the tricks I gave you, that you first simplify the question using your own words. Then after simplifying it, then you go back to the real biology. So that is now for part B. So that question is not so difficult as may seem to appear. So in this case, a neutrophil, you'll take it as if it is the bacteria. Phagocytosis, how does it work? Of course, bearing in mind that neutrophils engulf pathogens in our bodies. So when you're describing, you don't say that the neutrophil engulfs the food particle. If you say engulfs the food particle, you are off. Why? For using a wrong choice of the word. Because neutrophils have to be specific. Neutrophils do not engulf food particles. They engulf foreign particles in our bodies, or they engulf pathogens in our bodies. So you explain it in that way. You explain it in terms of phagocytosis in amoeba, but knowing what a neutrophil is and what a neutrophil does. It is a type of white blood cell. Then compare. Now, questions calling for comparison in biology, they need differences and similarities. And when you are writing these answers, write similarities, outline them. Then after that, write differences and also draw a table and give us these differences. Don't mix up points. You find point number 1 is a similarity, point number 2 is a difference, point number 3 a similarity. They will cross everything. So when they say compare, give both similarities and differences, but rate them independently. Okay, then part D, explain the sequence of events that occur when a plant cell is placed in a weak solution and a strong solution. Now here, using the word weak and strong, these are substitutes for dilute and concentrated. These ones here, they are substitutes for dilute and concentrated. So the word weak means dilute and strong means concentrated. Now here, I'll also give you another formula that we use here. When they say explain, they want to rate everything that happens. They're saying explain the sequence. Now here, this is what your explanation should capture. 1, when you're answering such a question, explaining events, or explaining practical or experimental results, because this is more of an experiment. It's like you get a plant cell, you put it in a dilute solution, then you see what happens. Then you have to report and explain your results. Now, when you are explaining, you start with what happened. You start with what happened, then you go to how and why it happened. This is the formula that we use. We use this formula when explaining experimental results. That is the formula that we use. And this is the formula that we can use here to explain, to answer this question of part D. So it means in your answer, I would be looking out for 3 things. Has the student told me what happened? Full stop. Has the student told me how it happened? Then has the student told me why it happens? Like that. Now, using this formula, I want someone to construct for us an answer for part Roman number 1. Don't be too wordy. Just a brief explanation using this formula here. Who is ready? Who is ready? Should I pick randomly? Okay, Vanessa.
Okay, let me try.
Yes.
A plant cell is placed into a weak solution which is dilute with a potential compared to that of a plant cell. It's going to create a water gradient is going to be created, which is going to be followed by an osmotic influx of water from the plant, from the solution to the plant cell, making the plant cell target.
Okay, now, Vanessa, are you still there?
Yes.
Now, in your explanation, you tell me what happened. We'll respond to this one. What happened?
There was osmotic influx of water from the solution to the plant cell.
Then how did that one happen?
It was by osmosis.
Pardon?
It was by osmosis.
Then why did it happen?
It was due to that the water gradient which was created, since the solution has a high water potential compared to that of the plant cell. Water gradient.
Now, Vanessa, now in your explanation, you have actually only told us 2 things: how it happens and why it happened. You have not told us what happened.
Okay.
So we shall see. I want another trial. Emma. Emma. Okay, let me have Trishila.
Teacher, for me, I think what.
Trishila, are you there?
Yes, you had muted me. Teacher, me, I think what happened is that the plant solution became turgid.
Is it the solution that becomes turgid?
Sorry, the plant cell became turgid.
How?
Because there was flow of water to the plant cell from the solution.
Why?
It was because the solution had more water molecules compared to the plant cell, and so they entered the plant cell.
And that is what the question wants. That's what the question wants. So realize that it would be easy to even rate your answer and check whether you have covered everything. And when you're writing, ask yourself what happened. It is even better to start with what happened. Then you go to the why or the how, like that. Is that clear? Ricky, anything to say?
No, it has been said.
Okay. Well, now that marks the end of our number one. These adaptations, you'll check they're straight in the notes. Now I want to go to number 2. Number 2. This one we shall also use 1 hour. Then number 3 and 4, those ones we'll try them out. Now, number 2 is a graphical question. Graphs. I had not to craft some of you. But however, I'm going to make sure that I give you tricks that you can use. I want to give you tricks that you can use to demystify or also to simplify these graphs. Let me first see what we have in the chat room. Okay, I'm reading what you wrote in this chat room. There is one, Priscilla Mutes is saying, what if I use hypertonic and hypotonic in my explanation? Now, if you use hypertonic or hypotonic, it is all right, but make sure you don't confuse those words. Always use words which are simpler. Always use words which are simpler. Someone is saying, pardon on the why part. No, the why part, now, the how part, so the what happened was the plant cell would become target. Then the how does it become target? Water enters into it by osmosis. Then why? Now the why part is the reason that the weak solution or the constitutional solution, so that the why part is that water will always move from a region of low concentration to a region of high concentration, or water will always move from a dilute solution to a concentrated solution inside the cell. Okay. So now, let me go to number 2. Now, number 2, if I mute these human beings here. Okay. Now, number 2 is about graphs. Now, whenever, actually, some of you, when you look at this graph, just a mere look at it, you tend to think that, oh my God, I'm finished. You become even scared, even bother to read it through. However, when you always pay attention to these graphs, you always find that actually they're the easiest to answer. Whether you know the biology or you don't know the biology, at least you'll find that you are likely, if you interpret it so well, you'll not fail to get more than 50% from that graph. Because realize that in most cases, only 30% is always about explaining the biology. 30% is always about explaining the biology. Then the rest of it is just giving us or telling us what you're seeing on the graph, the description or the interpretation or the comparison, those ones there. Now, when you look at a graph like this one, never rush to read these questions here. Never rush to read them. The moment you see this question, first close these questions, hide them, read the information given, and make sure that, okay, Alan, okay, I'll come to you later. And make sure that you have understood what that graph wants before looking at the questions that you're going to answer. That is the most important thing that you should always do. Put yourself in the shoes of the setter. Put yourself in the head or in the mind of the researcher, because all these graphs you're seeing here, they do not come from heaven and fall. A person like you goes out there, looks at how things are happening, gets information, puts it on a paper, and now attempts to explain why. Why are things happening the way they are happening? Because of this and this. And where are those answers always coming from? Those answers are coming from nowhere apart from your mind, from your head. You think in terms of biology and come up with the best explanations for them. Now, we're going to see. Now, let us look at this one. Let us interpret it together. They are saying, figure 1 shows the growth of a single plant leaf. Now there you first put a pause there. You ask yourself, what is growth of a single plant leaf? You interpret section per section. Why do you take time doing that? You want to make sure you understand this experiment. It is after you have understood it that you'll be able to get explanations that you'll be able to give, that you'll be able to use to interpret that data. Now, so here we are looking at one leaf, just one leaf like that. Then we are asking ourselves, how is this leaf growing? So we have one leaf. Then they are saying growth shows growth of a single plant leaf with its import and export of sucrose and phosphorus. Wow. During a period of 40 days. What do they want here? So they are saying, so we have our leaf. Let me draw it. And these things you should do if you see things are getting tough, if possible. So we have this one leaf. Then they are saying, we are looking at its import and export of sucrose and phosphorus. Import, we are seeing how this leaf is bringing in. So this is import, how this leaf is bringing in things. Then export, things are leaving this leaf, export. So we're looking at this leaf, the rate at which it is bringing in sucrose and also taking out sucrose, and also how it is taking out sucrose and taking out phosphorus. And remember, we're looking at a growth of the leaf. So it means this leaf starts when it is young, then it keeps on growing. So for us, as it is growing, you're going to see how is it bringing in things and how is it taking out things. People still have yes, Joanna. Any problem? Joanna?
Pardon, sir.
Pardon on what?
That importation and exportation of both sucrose and phosphorus in a leaf.
Okay, remember I'm using these arrows here. So we are saying export is taking things out of this one leaf. Then import is bringing things into the leaf. And which things are being brought in and out? Sucrose and phosphorus. And they monitored this for a period of 40 days. So that is how they did the experiment. Are we now getting into the mind of the researcher?
Yes.
Now, having got that interpretation, then now we can now look at the graph itself. What is the graph saying? On the graph, we can see, so here we have, on the vertical axis, we have import and export. So from 0 upwards, this is import. Import is that things are coming in. Then 0 downwards, we have export. Export is things are going out of the leaf. Things are being exported out of the leaf. So we are looking at the leaf as a country. We are looking at this leaf as a country. Okay. Now, let us interpret this question, this graph here. So what are we seeing? We are seeing that let us first look at leaf area. So leaf area is this. Then we have edge of leaf in days. The edge of leaf in days. So of course, when you look at the leaf area, what are they saying? They are saying, of course, with the increase in number of days, the leaf area increases. And then later it remains constant, which is obvious. You expect as the plant is growing, as the leaf is growing, its area must increase. And then it reaches a time when it stops, when the rate of its increase or when the rate of its growth pauses. So it remains at the same size. And that is what happens to leaves on trees. When the plant is still young, these leaves increase at a very high rate. Then when the plant reaches a certain age, the size of the leaf remains. So that one is obvious. Then now let us look at another concept. Let us look at now phosphorus. What are we seeing on phosphorus? We are seeing that from zero here to 20. From 0 to 20, the amount of phosphorus imported increases. The amount of phosphorus imported increases. Then
gradually
from 20,
or rapid?
Now is it gradual or rapid? Now here we shall use this one as a comparison here. Do you think this is a gradual or a rapid change?
It's a gradual.
Gradual.
This one obviously is gradual.
Gradual.
Then again, we can see that from 20 to here, the amount of phosphorus imported decreases gradually up to 0. Now, we have to identify which day is this here. Remember, the moment we cross this line here, this horizontal line, downwards, it will no longer be import, but it will be export. So it means from this point here up to here, the amount of phosphorus exported out of the leaf. Now, is it increasing or decreasing?
It is increasing.
Increasing.
Is increasing.
Yes.
Then, from this point to this point, what is happening to the amount of phosphorus being exported?
Decreasing.
Gradually.
Is it decreasing or increasing?
It's decreasing.
Very slow.
Yes.
Excuse me, teacher.
Yes.
Excuse me, sir. Teacher, could you please help me? Because I get confused, like when to say gradual, rapid, like when do you say gradual, when do you say rapid? When describing a graph. And how can you know that this is gradual?
Well, you just use your you just use your eyes. You look at the steepness of the graph. You look at steepness of the graph. And I hope when I say steepness, no one is getting lost. So you look at the steepness of the graph. Where the graph has a very high steep, then that is rapid. Where the graph has a low steep, then that is gradual.
Excuse me, teacher.
Yes?
Why I thought we do not use the vertical axis when we are describing.
Obviously, when we are describing, we never quote any value from the vertical axis. We don't quote.
So why did you use at zero?
Well, I'm still using it in my interpretation. Remember, I'm just taking you through how to interpret. So when I was interpreting, I mentioned this zero to let you know that I'm meaning the point where this line has touched here, zero. So I'm still using it in my interpretation, not in my description. Okay, now, someone was saying that how do we know that it is gradual or rapid? Now, when you look at this line here and compare it with this one here. So obviously you can see that this one here, the slope is steep. So this is a rapid increase compared to this one here. So this one here is a gradual increase. And the word gradual means the same as slow. So you can either use the word slow or use the word gradual. Okay, hope that one is clear. Someone is saying that, excuse me, teacher, on which graph do you use exponential? No, you avoid using that word exponential. Just use the word rapid. Actually, exponential means rapid. So avoid using the word exponential, just use the word rapid. Then Namoding is saying that since the question is asking for description of the import and export of phosphorus, so do we consider each graph independently? I.e. the rapid for one graph can be different from the rapid of the other. Now still here, you just use your common sense. You just look at them, you see where there is gradual or rapid, but you must make a fair judgment. It must be a fair judgment. Now, for instance, when you look at this graph here, look at this now, look at this decrease and also look at this decrease here. Then look at this increase here and also look at this increase here. Obviously, you see that there is a difference. You can see that this one here is gradual and this one here is rapid. You can judge. You can judge. So when you look at this one and you compare it with this one here, obviously you can see that this one is rapid and this one is gradual. Okay. Okay, then yes, Priscilla, ask your question. Priscilla, we can't hear you. Apollo, ask your question.
Thanks, teacher. What about when you're comparing between the curve for phosphorus and that one for sucrose? How do you compare them?
We shall look at the comparison later. Wilson.
Thank you, teacher. Now, can we use words like less something or more when it comes to description?
Now that is a good question. Can we use words like less or more? Now, we use words less or more only when we are comparing. We use the word less or more only when we are comparing. I think now this one also includes Apollo's question. We use the word less or more when we are comparing. For instance, if they ask you to compare, now I want you to look what is happening here in this region here. Now I want you to look at the graph. Let me use the ones where we have the same scale. Look at the graph for sucrose and phosphorus in this region that I have circled here. This one here is phosphorus. This is phosphorus. Then this one here is sucrose. Now, they want you to compare the two there, those two changes. Now, in this comparison, first of all, when you look at all of them, the increase is gradual. The increase is gradual. But one is more than the other. So I can say that the graph for sucrose is more gradual than the graph for phosphorus. So that is when we use words like less or more. If you look at, let me use maybe, if maybe I had another graph here like this. So I want to compare this one with this one here. So if this is A, then this is B. So obviously, what does it mean? I will use the word more or less. I can say the graph for B increases more rapidly than the graph for A. Meaning that it means that, first of all of them are increasing rapidly, but one is more rapid than the other. So don't use those words less and more on one graph. You say that, for instance, that here it is less rapid. Then here it is more rapid. Then here less rapid. No. You use those words when you are comparing two graphs.
You guys are making me noise.
Okay. Then Gloria describing the negative part, we shall come to that one. Do we use almost constant or fairly constant? Using the word almost or fairly. Now I think, first of all of them are correct, but almost sounds better. Using the word almost constant sounds better. Then ethyl. So teacher, for this case of comparing, does this mean that we don't need any biological explanations, more elaboration? That is a very good question. Let me read this question again for all of us to understand. It's saying that for this case of comparing, does this mean that we don't need any biological explanation? Well, now, in biology, when they ask you to describe a graph, when they ask you to compare, they don't need any explanation. They don't need any reason as to why. You just see what is on the graph and you just tell us what you are seeing. So that is what they mean by comparing and describing. No reason whatsoever. You just tell us what you're seeing on the graph and you go with that. You don't give any reason. Then teacher, what of relatively? Now, avoid using, now of course, relatively also avoid using it. Relative is like a comparison, relative to this. Don't avoid using those other confusing words. If it is a gradual increase, say gradual increase. If it is a rapid, say rapid increase. Also, avoid using words like slight. Avoid using them. But however, there are some unique graphs where you find that you are almost tempted to use that word slight to make your description stand out. So those graphs can be there. And because of time, we shall not go into those examples there. But just know that there are some unique instances, like when you feel like you have to use the word slight. But under most circumstances, avoid using that word slight. Because I'm sure when you feel like using the word slight, it means the change is gradual. So instead of using the word slight, use the word gradual. What if they ask to explain the relationship? That is a very good question. If they ask to explain the relationship, now the relationship, there is an aspect of comparison. Then you explain them together. For instance, if they ask you to explain the relationship in this scenario, they can ask you to explain the relationship between leaf area and the phosphorus. They can ask you to explain the relationship between leaf area and phosphorus import. Or not even import, just they can ask you to explain the relationship between leaf area and phosphorus. Now, if they ask to explain leaf area and phosphorus, now, first of all, you first, when they ask to explain a graph, you start by describing. Then after describing, you give the explanation. Now, if that question is saying that explain the relationship between a leaf area and phosphorus, you have to first describe for us that relationship. Then after describing that relationship, then you give us the explanation for that description. I'll give you an example here. So let me use, so we have leaf area and sucrose. So we can see that as the leaf area is increasing from 0 to 10 days, the amount of phosphorus imported, there is an increase in the importation of phosphorus into the leaf. That is the relationship that I'm seeing there. Maybe let me type it so that it can make sense, because some of us, we understand better when we see things. So I'm saying, so I want to answer that question of explain the relationship between sucrose and the phosphorus. The first thing to do, I have to bring out what is that relationship that they are talking about. I can say that from 0 to 10, there is something. Then from 10 to 20, there is also some change. Then from 20 to around 27, there is also some change. So let me start with this one here. So I will say that from from 0 to 10 days, both you can say both importation okay. Both importation of phosphorus, and the let me say from 0 to 10 days, there is a gradual increase in the importation of both phosphorus and the sucrose. So that is the relationship. And this relationship is a similarity actually. So we can see that from 0 to 10 days, there is a gradual increase in the importation of both sucrose and phosphorus. Now there, I have described a relationship. What is the next step? Remember, we are explaining. Then now I have to give an explanation why. Why is it that as the number of days increase, there is also a gradual increase in importation of both phosphorus and sucrose? Then I can also go on and say that from 10 to 20 days, from 10 to 20 days, which relationship am I seeing there? The importation of sucrose, increases rapidly, whereas
from 0 to 10 days,
whereas the importation of phosphorus, increases gradually. Because now here, of course, when I'm giving when I'm describing my relationship, I don't have to put the word because, but I'm putting the word because I'm trying to answer different question of explaining. So this is how we would pick out the relationship. You first pick out that relationship and then explain it. Then I can still go on and say, now from 20 to 27 days, there is a rapid increase in the importation of sucrose, whereas the importation of phosphorus is decreasing gradually. As simple as that. So that is how we would give the relationship or describe the relationship. Now, suppose the question was saying, compare the changes of sucrose with the changes of phosphorus. Compare. Remember, I told you how we answer comparison questions. You give similarities, you write the word similarities, then you pick out only the similarities. Then after, you give difference, you write the word differences, and you pick out differences. I saw a question in the chat room. Do we give differences in a box format, or we just outline statements? Whichever way is easier for you. When you're giving differences, even of graphical data, if you see that it is easier for you to draw a table, draw it. If you see a table won't be the best option, you can use words like whereas, while, as, like that. You're free to do what is convenient for you. Now, let us develop maybe an explanation here. Yes, Louis?
Chair, I was kindly requesting, Chair, I did not get the question because of my network, but may you please help me the question? I'm writing, but I don't have the question.
Explain the relationship between phosphorus and sucrose. Is what we are answering. The answers I'm writing here. Explain the relationship between phosphorus and sucrose. Okay. Priscilla. Priscilla, are you there?
Yes, teacher.
Your hand was up, Priscilla. You have a question?
Yes, sir. Couldn't it be better to write from 10 days to 20 days rather than just writing from 10 to 20 days?
Aha. Do we start from a bigger day to a smaller day, or we start from a smaller day to a bigger day, number of days?
We start from a smaller day. No, my issue is with this from 10. Okay, there is no the word days, and it's saying from 10 to 20 days.
Oh, okay. So here you say we can say from 10 days to 20 days. Yes, it is.
Yeah, I'm just asking, the most correct isn't thing. It better to write from 10 days to 20 days rather than just writing 10?
Yes, you have to attach units to every figure that you write. So here I skipped the days, but it is better for every figure that you write, attach there its units. Yes, Beno?
Okay, sir.
Teacher. Okay.
Yes.
Saying that graph for phosphorus means from 10 to 20 days, it's being exported rapidly. That's what it means.
Beno, I've not heard anything. The network was not clear. You type your question in the chat.
Okay.
Okay, so Veta is saying, what if I don't include gradual or rapid, but just say decrease or decreases? Now, the word gradual and rapid, those ones are descriptive words. You must use them. If you don't use them, you lose some marks, because you find the marking guide would have attached marks there, that if a student says gradual, you put a tick. Rapid, you put a tick. So for if you just say it increases or decreases, you end up missing out on some points. Then Arnold Johnson is saying, sir, I think it is very appropriate to present the differences in tabulated form. Now, tabulated form. Now, for questions which come from areas, okay, for non-experimental results, questions, like for example, if they say, give differences between maybe osmosis and active transport. There it is better to use a table. But for graphical data like this one here, please use what is convenient. From experience, I have seen some differences of graphical data where it is almost impossible to get to use a table. Impossible to use a table format. Very impossible. You find that using statements would be the most convenient and appropriate way. So don't limit yourself. Don't tell yourself that you must always use a table. But, and don't quote me wrong, in most cases, a table is better. So always first think in terms of a table. When you see that a table may not be appropriate or a table may limit you, especially for graphical data, then there you can use words.
Excuse me.
The relationship.
Yeah, but now if you are to tabulate and you are to answer, use of hyphen, is it allowed?
Now use of hyphen. Now for us in biology, we are not after proper use of which English have you used, which word have you used. For us, as long as our answer is flowing, whether I have used the commas, whether I have used the full stops, whether I have used the hyphens, as long as our answer is flowing. For us, we don't care about those symbols that you use. For example, you look at this first point here, where you're saying that from 0 to 10 days, comma, there is a gradual increase. Now, another person, instead of using a comma, I can even say, full stop. From 0 to 10 days, full stop. Continuation, it is a gradual increase. In biology, it is correct. Because I will say, has the student quoted the correct number of days? Yes, I put a point there. Then has he described? Yes, I put a point there. It is correct. Another person can decide to put there semicolons. It is correct. For us, those points do not matter to us. What matters is the flow of your statement. Okay. Patricia?
Yes.
Patricia, are you there?
Yes, teacher.
Your hand is up. You have a question? Okay, Angela Kakunguru. Angela, what's the question?
Teacher, I think you made a mistake. From 10 to 20 days, the importation of sucrose actually decreases.
Yeah. So from 10 to 20.
Yeah. It's the export that then later on starts increasing.
We're having a different graph. Sucrose. Oh, I was following the line for leaf area. I follow the line for leaf area. So it is from 10 to 20 days, the importation of sucrose
decreases.
So again, sucrose. So it is from, actually it is from 10, not even to 20.
To 11.
Yes, from 20 to 11.
Yeah.
Importation of sucrose decreases. Like that. But however, what is the point here? Have we learned how to give or how to explain such questions that are asking for relationships? Now, Maybe let me show you that we can also think of an explanation, even if we have not covered that area. Now, let us develop an explanation for this one here. So here we are seeing that from 0 to 10, as the leaf is growing, sucrose and phosphorus are being imported into the leaf. Now, the first thing you have to ask yourself here is, why does this growing leaf need sucrose? Or more so, does it need sucrose? Can someone answer me? Does the growing leaf need sucrose if it is carrying out photosynthesis? Yes, Joshua?
It may be
Louis. Louis,
yes,
pardon?
teacher.
Yes, teacher. It may be needed later on,
Why?
even in the
it's needed.
Did the Calvin cycle, Hussler cycle, something like that, but it's needed. It's needed during in other cycles as well. So sucrose is very needed.
Okay. So now here it means that, remember, sucrose, first of all, leaves make sucrose during photosynthesis. But however, this leaf is still young. If it is still young, it means that most of the sucrose it will need, it is going to be imported, brought to it, because it is still young. The rate at which it is carrying out photosynthesis cannot produce enough sucrose that it requires, even given that its surface area is still small. Then you see that later, when this leaf matures, actually, most of the sucrose is exported. Why? Because it means that now the surface area of the leaf has increased. This leaf can now trap a lot of sunlight energy, carry out a lot of photosynthesis, make a lot of sucrose in excess that it even ends up exporting some out of it. Then now that is the reason behind this decrease, behind this increase in the exportation of sucrose.
I beg your pardon?
I've said that the reason why at first we see that this leaf is bringing in sucrose, is importing. The reason why this leaf is importing sucrose is because it needs sucrose to carry out metabolism. To carry out metabolism that will result into its growth, the growth of the leaf. Why is it not relying on the one that it is making itself? Because we know the leaves are supposed to carry out photosynthesis and produce sucrose. This leaf is still small. Its surface area is still small. Therefore, the amount of sucrose it is making is very little. That it mainly relies on the sucrose that is being imported into the leaf. Then you can see that after 10 days, now this leaf now starts to export. The one that was importing now starts to export. Why? It means that at this time, the leaf has matured. The leaf has now enough chlorophyll to trap enough sunlight energy, and it is making a lot of sucrose than it needs. Therefore, it has to export the excess sucrose. That's why you can see that from 11 to 30 days, the amount of sucrose exported increases rapidly. Then, what of the phosphorus? Why is it that when this leaf is still growing, you can see for phosphorus, it increases, it exports more from 0 to 20. So in other words, it imports sucrose when the leaf is still young, so the phosphorus. So you have to ask yourself, why does this leaf need sucrose? Sucrose is needed to make ATP. Sucrose is needed to make ATP. Also, sorry, phosphorus. Also, this phosphorus is needed to make some of the components in the plasma membrane of the leaf cells. Remember, as the leaf is increasing in size, new cells are being formed. And the plasma membrane or the cell membrane of these leaf cells, phosphorus is a constituent. So it means this leaf needs phosphorus for two things. One, synthesis of ATP. Then secondly, formation of cell structures. Cell structures like the plasma membranes. That's why you see that as this leaf matures, it also actually starts exporting the phosphorus. Why does it export the phosphorus? Because it has now matured. It is not forming any new cells. So all the phosphorus that is coming into it ends up being exported. Remember, this phosphorus is not made by the leaf. No. This phosphorus is absorbed from the soil. So as this phosphorus flows into the leaf, all of it, or most of it, is exported out. Why? Because when the leaf is very mature, it is no longer forming any new cell parts or any new cell structures. When it is young, many cells are being formed. Therefore, that's why you see it imports in a lot of phosphorus. Then why is it that after 30 days, after 30 days, the amount of sucrose imported decreases? Why is it that? By the way, when this curve is going up, this is a decrease. When it is going down, this is an increase, because we're talking about the negatives. Here, we're talking about the negatives. It's like if you flip this graph and if you flip it, it's like flipping this graph here to put it up. It will still appear like this. If you flip it to put it up, if you flip it, this one, it will be like, it will appear like this. So that's why we're saying this is an increase. This is an increase. From here, it is increasing. And here it is decreasing. We're putting into consideration the aspects of these negatives here. I hope that one is clear. So why is it that after 30 days, here, we see that the amount of sucrose being exported, export means coming out of the leaf.
Excuse me, teacher.
Okay. There is a question.
I don't really think, because I don't think that it is like that, because you said as the leaf keeps maturing, it will need to export the sucrose. So if we are saying that up curve is decrease, as you say, then won't it mean that the sucrose after the leaf has matured is decreasing? Oh, wait.
Well, now this is what, maybe let us first look at the explanation. What would happen when the leaf matures? Now, at this level, after 30 days, this leaf has matured. After that days, the leaf has matured. Now, when the leaf matures, the rate of photosynthesis in that leaf decreases. Why does it decrease? When the leaf matures, it starts to lose chlorophyll. It starts to lose chlorophyll when the leaf matures. That's why you see mature leaves start to turn yellowish in color. The reason why they turn yellowish in color is because they are losing chlorophyll. So as they lose chlorophyll, you also expect the amount of sucrose made to reduce. So if the amount of sucrose made to reduce, so it means even the amount of sucrose that this leaf was exporting, the amount of sucrose that this leaf was taking out reduces. Why? Because of reduced rate of photosynthesis. Why does the rate of photosynthesis decrease? Because the leaf is maturing. Because the leaf is now losing chlorophyll pigment. Louis, is it making sense?
Teacher, it's making sense now.
So it means that when the leaf has matured, the amount of sucrose it exports decreases because it is making little, it is making less. But here, when it is still in its youthhood, when it is still growing rapidly, the rate of photosynthesis is actually at its maximum. It is carrying out a lot of photosynthesis, therefore making a lot of sucrose. That's why here you see the amount of sucrose being exported increases rapidly. Okay. Aaron, Noah? Aaron, are you there? Ediango?
Excuse me.
Yes.
Yeah.
Okay.
Teacher.
Hello?
I'm listening. You can speak.
Okay, I'm like, I wanted to comment on the decrease in the decrease in the uptake in the export of sucrose. That at old age there is a enzymatic breakdown of chlorophyll, which reduces its photosynthesis.
Now, the chlorophyll is not broken down by enzymes. I don't know where you read that one from, but the chlorophyll is not broken down by the enzymes. So you just simply say that there is a loss of chlorophyll pigment as the leaf matures. You don't go on to give us other details that may not be scientifically correct. Okay. Yes.
Like, there is something I'm not getting clear about exportation. Why are we considering the first change as decrease, like as in increasing? Why are we considering it as increasing?
Oh.
Same.
Well, now, why are we saying that this one? Now, why are we saying that this one here is increasing? Now, if you have a date, because minus means a date, if someone has a date of 0, then another person has a date of 2000. So, which of these people has a bigger date? The one who has a bigger date is that person of 2000 shillings. So it means as we move from a date of 0 to a date of 2000, your date is actually increasing. So when your dates reach 2000, then you can now start decreasing your dates. You want to take them back to 0. So meaning that now as you move from your date negative, you go to 0, then that is a decrease. You are decreasing your date. Negative means a date. Is it coming somehow clearer?
So teacher, you don't take this concept of saying negative one is greater than.
It is true, but remember minus. What is the meaning of the word minus? Minus is a debt. If you have 1,000 in your pockets, and your neighbor, they are demanding your neighbor 1,000. So they are demanding your neighbor 2,000. Who has more money? Is it the neighbor whom they are demanding 2,000, or you who has 1,000 in your pockets? Obviously, for you, you are in positives, so you have more. Then the one who is in negatives has less. So if he also tries to move towards you, then he is decreasing his debt. So that's why we are using this one as an increase, then this one as a decrease. Okay, there is a question here. Someone is saying that, teacher, is it allowed to approximate values that are not given? Like in that case where you assumed 10 to 11. Wow. Now, 11 is not that 11 is not given. 11 is there, but the graph was drawn to a scale of 10, 10, 10. It doesn't mean that there are no figures in between here. The figures are there. And actually, when you're quoting figures from this horizontal axis, you must quote them accurately. You must estimate them accurately. If you want to mean this point here, you have to give us the real value that is here. You have to estimate it. If you want to give us a value here, you have to estimate it and give it to us accurately as it is. Okay. Someone is saying that what causes, yes?
Master, sorry to take you back, but there's something still confusing me. On relationship, do you give only the similarities, or you can give both the similarities and the differences?
Now, did you read the points which I had written here?
Yes, but one was a similarity, another one was a difference.
And that's how it is. Have I answered you?
Yes.
You give both.
Excuse me, sir.
Yes.
Yeah, sir, there is another observation I've seen here from.
Give it to us, please. Irene is saying, how can I estimate? Now, that's where you have to use your science. That how can I know that, for example, this one is 27 and this one is 11? When the graph is not plotted on the graph paper, you use your common sense. If you have to get a rule and put it there on paper, you have to use whatever means and estimate and quote for us that value accurately. Okay, so we shall never finish. We shall never finish. Because when we start talking biology, biology never ends. And I think, okay, Cosima is saying now teacher means relationship and compare are the same. Yes, relationship and comparison are the same. So I think because of time, I believe that we have acquired a lot. We have at least got some skills. Of course, there are some things that we may not have talked about, but I'm sure we have talked about 90% of the things that will help us. Kitty Brand is saying explanation for phosphorus on maturity, please. Now, phosphorus on maturity, so the phosphorus on maturity means now when the leaf is mature, this leaf, remember phosphorus is needed for two things. Phosphorus is needed for phosphorus to make ATP, And at the same time, phosphorus is needed to make new plant cell walls or cell membranes. Now, when the leaf matures, it stops growing. It stops growing. And also, the rate at which it is carrying out photosynthesis reduces. So that reduces its demand for phosphorus. It reduces its demand for phosphorus. Why? Because it is carrying out little or less photosynthesis. And secondly, it is no longer forming new plant cell membranes. Therefore, most of the phosphorus that enters the leaf ends up being exported because it is not used. The leaf is now carrying out less photosynthesis, so it does not require a lot of it. And at the same time, it is no longer maturing, so it is no longer growing, so it is no longer forming new cell membranes. That is the explanation for phosphorus. Someone is saying, I mean, import and export differentiate. No, import and export, this one were just used here for this case. And when you're starting this lesson, this graph, I told you the meaning. Then I just wanted to comment that sign on the graph by that it is export. Yes, the negative sign was just implying that it is export, that the leaf is losing something. That's what the meaning of the word negative sign, that the leaf is losing something. When you talk about initial in the graph, now you use those words of initially when they have not given you figures on the horizontal axis. When they have not given you figures on the horizontal axis, then that's why you can use words like initially. Like for example, if we didn't have any figures on the horizontal axis, we would have said that initially, the amount of sucrose and phosphorus imported increases gradually. Then later, it increases rapidly or gradually, like that. So we use the words like initially when we don't have any figures on the horizontal axis. Then what happens to phosphorus after 40 days? After 40 days, what happens to phosphorus? Here the graph is showing you, it is now, what it means is that when this leaf receives phosphorus from the soil via the xylem, because it comes with the water, if it enters that leaf and it is not used, it leaves and goes perhaps probably to other leaves that need it. So it is export means that these materials are leaving the leaf and going to other parts of the plant. That is what export means. Then what about when I say that photosynthesis makes glucose, which is converted to sucrose, which is the end product of photosynthesis? It is transported the phloem, but accumulation of sucrose delays flowering. Gloria, no, please. Accumulation of sucrose does not delay flowering. That is wrong biology. Then Kule is saying project for us, the questions on the graph. The question on the graph, of course, I don't know. I'm going to share this document in our WhatsApp group. Now, those ones who are not in the WhatsApp group, you pick my number from the inbox, then you inbox me, then I add to that group, then I'm going to share there the solutions to these questions here for your further reading.
Excuse me. Excuse me, teacher.
Yes, please.
I had some question about the amount of chlorophyll, which you say that it reduces when the leaf matures. Now, why does it reduce when the leaf matures?
Because the leaf is no longer growing. The leaf does, if it is no longer growing, it doesn't need that phosphorus and sucrose as well.
But if I give answers like maybe when the leaf matures, the cells are worn out.
No, the cells are not worn out.
Just because the leaf stops growing.
When the leaf matures, it means it is no longer growing.
Okay.
Stops growing. You don't say that they are worn out. No. Someone is saying, how about after 35 days, the phosphorus export seems to decrease? Yes, after 35 days, the phosphorus export decreases. Why? It means that even very little phosphorus is entering this mature leaf. And eventually, this phosphorus might even go to zero, meaning that now the leaf is not importing and not exporting anything. That is when now the leaf is completely dead. When the leaf completely dies, it no longer imports phosphorus. And if it no longer imports phosphorus, then it even means it will not export it because it exports what it has imported. Okay, so I think please let us end the lesson from here. Thank you for being good students. Please pick my number from the chat. You WhatsApp me, I add you to the group.
For
I
being
am
a
going
good
to add.
Teacher,
Yes?
that's the importance of phosphorus.
It is used in the synthesis of ATP. When these plants are carrying out photosynthesis, they generate ATP. And remember, ATP is adding adenosine diphosphate to a phosphate group. So that phosphate that is added is the one that is coming from the phosphorus. So the importance of phosphorus to a plant is to make ATP and also to make cell membrane components.
Teacher.
Yes?
Teacher. Now, if you talk about the comparison
we are not clear.
What did you comparison? If they have not as comparison, the relationship, master.
I have not got your question. Please, time, we are out of time.
What I mean,
you look at the differences, sorry, the solutions.
Like if you ask the comparison and the similar, and the relationship. The relation and the similarity.
Okay, let me wish everyone a nice day.
Master. Master.
Bye.
Master. Master. Hello, Master. Master.